Many challenges can stall in advanced therapies development, often tied to inadequate preclinical planning and process development. Now, drug developers can prevent issues before they arise and avoid the rework and delays that can negatively impact timelines and derail development. By incorporating best practices such as design of experiment or DOE, process development can be streamlined and optimized using systematic methodology to estimate variable interactions and thereby reduce the number of experimental trials required. And notably, DOE can have a direct impact on process development for the complex manufacturing projects in AAV gene therapy applications. I'm Miles Skoda, and this is the Life Science Connect webinar series, and today we're talking optimization by design, a critical factor in successful viral vector scale-up. Now, joining me for this presentation are two industry experts. First, Andy Morio, Head of Process Development for Andelin Biosciences, and Paul Giroux, Scientific Support Specialist for PolyPlus, who together will discuss key considerations for taking your advanced therapy from concept to commercialization with confidence. Now, we'll be holding a Q&A session following this presentation, so please feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we don't get to, we'll follow up on after the event. And then just a bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, submit a question via the Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day or two, and you'll get an email with a link to it, or you can just use the same link that you used today. So with all that said, I'm going to turn it over to Andy and Paul to get started.
Thanks, Miles. So yeah, to talk about our agenda today, we're going to start with some quick introductions to Andelin and PolyPlus, and then go into AAV manufacturing platforms and limitations of plug-and-play approaches, and then the benefits of DOE experiment approach, and then walk through a couple selected case studies. And then talk a little bit about, how we're leveraging partnerships to expedite process optimization. So, introduction to Andelin. We always start with, always like to start with our mission and vision. Being born out of Nationwide Children's Hospital, I think one thing that sets us apart from any CDMO really is our connection to the patient, and that's directly reflected in our mission and our name, which is derived from Andrew Kilbarger, who at 8 years old received the first U.S. investigational drug for Duchenne muscular dystrophy in 2006. And then Evelyn, who at 8 weeks old received the first experimental gene therapy drug for spinal muscular atrophy that directly led to the Zolgensma product. So being spun out of Nationwide Children's Hospital, we brought 20 plus years of expertise in the personnel and in technical know-how, and then we also added a strategic partnership and investment with Cytiva, and we have 20 plus years of gene therapy experience. 450 plus clinical batches under our belt, as well as 75 plus INDs. Currently in our new facility, we've undergone 9 successful audits with client approval, two successful QP audits, and then we have 2 active BLAs in preparation. And talking a little bit about our facilities, we have 3 facilities in the greater Columbus area. There's, a joke locally that says everything is 20 minutes, and that's definitely true with all 3 of these facilities. They're all very close together. Our viral vector Andelin Development Center is located in Dublin, Ohio. That's where I'm sitting today. It's a 42,000 square foot facility that handles all of our preclinical and development from PD to AD and our preclinical manufacturing efforts. Then we have our Andelin Corporate Center, which is a fully commercial GMP facility that's 180,000 square feet, that's able to do suspension manufacturing up to 2,000 liter and adherent 2D adherent manufacturing up to 60 hyperstacks per sublot. We can manage multiple sublots at any scale. And then our plasmid facility, which is 9,500 square feet, that handles both GMP and non-GMP manufacturing, it scales up to 200-liter fermenters, as well as cloning services and in vector design. So what we've done here is built a concept to commercialization CDO with total vertical integration from starting materials through manufacturing, development, fill finish, and as well as analytics. We've brought plasmids in-house on the front end and brought most of the analytical methods in-house on the back end in an effort to control quality as well as control timeline. So the idea here is any client could come to us at any point of entry in their product life cycle, and we could hit the ground running. So with that, I'll let Paul do an introduction to PolyPlus.
Thank you, Andy. So many of you may know our products, but not all of you may know PolyPlus, so I will do a quick introduction of the company as well. So we are a French-based company in Strasbourg in the east of France. So for a long time, we've been known for transfection reagents, being a pure player in transfection for quite some years, as we were funded by the actual team that published the first results on and patent on using PEI for transfection. It's been more than 20 years though since the creation of the company, and we've grown a lot and implemented a lot of other solutions to answer to our gene and cell therapy customers. And as you probably have heard, the biggest news is that since this summer we are now part of Sartorius. So we have different facilities in Europe, so in Strasbourg, but also in Lyon and in Lille in France, but also in Liège in Belgium. We have a sales team all over the world to support our customer, and we now provide solution in nucleic acid delivery, but also plasmid engineering, molecular and biology services. We also perform plasmid and protein manufacturing up to the GMP level. We also provide GMP fill and finish capability. And all of that is possible thanks to our end-to-end capability in product and service development, process development, manufacturing, quality, and most importantly, regulatory and scientific support. So as I was saying, we started as pure player in transfection, but nowadays we are more solution provider especially within the upstream space of cell and gene therapy. Hence, we expanded our portfolio over the year up to the point that we were bought by Sartorius to have really a complete portfolio of products and services to answer our customer need. But it's not just about the product, which need to be excellent and of high quality, and that's why we are in the GMP space heavily as well. But also through what I do on a daily, day-to-day basis, which is scientific and regulatory support to our customer to help them move forward. We are also involved in the mRNA and DNA therapeutic market through non-viral based delivery, but that won't be the topic of discussion today, as we will focus mostly on AAV manufacturing. And with that, I hand it over to Andy again.
Yep, thanks, Paul. So talking a little bit about limitations of plug and play approaches. So, just a little background to start is, even with the current economic climate, trends and numbers of clinical trials, products being registered continue to grow and grow in an exponential fashion. And then on top of this, if what you consider that, as AAV mediated gene therapies move out, well, not out of, but beyond the rare, ultra rare state into more standard of care, that the amount of demand for AAV is going to explode as in terms of number of trials, number of indications, as well as dosing. Dosing continues to increase in terms of number of VGs dosed per treatment. And then all of this leads into not just scale issues of how much vector needs to produce, but it's also the diversity of AAV vectors is poised to continue to increase with more and more engineered capsids being developed for specific tropisms. The DNA cargoes are becoming more and more diverse, and as each of these continue to differentiate, the interactions of the two also continue to differentiate, which creates more and more demand on the downstream purification in terms of flexibility. So, all of these questions end up coming into the fold of how big is the patient population, all the traditional questions, what's the indication, delivery route, all of these things lend themselves to increased demand, but it also makes it so not one size fits all. We meet that demand of scaling up and scaling out very neatly with with a platform approach, because this streamlines cost of goods, it streamlines production, but there's some pitfalls in a plug and play approach that we have to address. As Andelin and as an industry, what happens is we become too rigid, and rigidity also reflects weakness in a platform. So we have to also understand where development is needed, where optimization is needed, as well as where modularization is needed in a given platform, whether that be in the transfection or in the clarification, chromatography steps, or final formulation, etc. There's multiple steps here, depending on the indication and the needs of the client, the needs of the patient, that may lend itself to the need for flexibility in the platform itself. So how do we do that, right? We endeavor to build a platform that's ultimately configurable by faster optimization and efficiencies, having different modalities. We also build an optimization toolkit, and the tools in that toolkit are built around, in many cases, DOE, and that is one of the most powerful tools we have in that kit to be able to provide flexibility and really go beyond scaling up or just scaling out. And all of it, of course, is data-driven, and this is where 20 years of experience and 450 clinical batches really becomes an asset, as we can lean on that data to understand not just historically what has happened, but use it as a predictive tool in performance going forward. So, Paul, would you like to continue by talking about benefits of DOE approaches?
Thank you, Andy, for this introduction on that. And I'm glad to say that actually Polyplus and Andelin are really aligned on that topic, that there is no one size fits all or one solution fits all solution platform to AAV manufacturing. And that's why we've been pushing more and more DOE, design of experiments, to help our customer actually test out different solutions and help them reach really new targets in terms of titer and scalabilities. So I'd like first to take a step back. You mentioned scale out versus scale up. I wanted to present actually the fusion that we believe are approach that people can take to meet the different approach people can take to meet the global demand in AAV, and to reduce the cost of goods, to also make this therapy available to more and more patients in the future. So we think there are three solutions. The first one would be scale out, which has been done historically, works really well. It was most of the time a straightforward solution. Basically, if you have a process that works well in small scale, you can basically just multiply the number of bioreactors you do. However, there is huge limitation of that, is that there is a linear increase in cost in terms of cost of goods, but there is also a linear increase in cost in terms of labor cost, but also in terms of equipment and facility costs. So the more you need to produce, the more place it takes, the more work it takes. So that's why more and more people have been focusing in the past few years on scaling up, actually. So moving from 10, 50 liter bioreactor to 500 up to the 2000 liter bioreactor. So it's basically we've taken with the gene therapy the same approach that people took many years ago with the protein therapeutics market. So increasing the volume to basically reduce the OPEX cost and decrease the cost per doses and allow you to produce more in one batch while still maintaining the same productivity. Now, scale-up is important, but we do believe that it will not be sufficient currently for the AAV market. So it's still in its infancy, so there is a lot to do on process simplification as well. And by process simplification, I mean basically optimizing our process to be able to produce from one cell more AAV capsids, but actually also more AAV in terms of quality. So we know full empty is a big topic that people are focusing on. Quality of the viral vector that is produced is not crucial, both for the safety of the patient and to improve the yield. But simply, if one cell can produce more AAV in the end, you will be able to produce more doses and treat more patients, and it will drastically reduce the cost. And how can you do that? How can you intensify your process? So there is a few solutions that Polyplus has been working on. The first one, that's our core expertise, will be transfection regions. So we try to develop a new generation of transfection reagents to meet this demand in higher titer, higher quality of products, but also in better scalability of this transfection region to allow customer to move more easily toward larger scale. But we can also work on plasmid engineering. That's a solution that is present at Polyplus nowadays. We are also working on optimized cell lines, so that could be either the cell line itself, but also everything that goes around it, meaning the medium, the feed, everything that you can use as enhancer of productivity or cell fitness. And all of that taken together can help customer achieve better results. But also what we are trying to push more and more is for people to adopt a design of experiment approach. And I will also talk about that a little bit later. First of all, I just want to make a little side step by talking about next generation transfection reagent and presenting VectorV AAV. So how VectorV AAV, when it hit the market in 2020, did help improve the capacity in terms of AAV production. So when we launched VectorV AAV, we saw that we could have an increase in terms of titer from 2-fold from PI Pro up to 10-folds to other competitor reagents. So we've seen a wide array of fold increase from people switching to VectorV AAV. So it has been a tremendous hit for our customer and a really good reagent. And basically, when you think about increasing the titer, you are actually decreasing your costs exponentially. Because if you take into account a large-scale bioreactor, a 2,000-liter bioreactor, if you want to produce a certain amount of doses to treat patients, and with a poorly adapted process, you need to produce, for example, to run 10 2,000-liter bioreactor to treat all of your patients. By having a 10-fold increase in titer, you only need to run one bioreactor anymore. So in terms of decrease of cost, that's tremendous. That has a direct impact to in manufacturing cost afterward. And as mentioned, we also believe that all of this come together, but you need some tools to actually make informed decision based on scientific data. And for that, we are pushing more and more, and this is something that I do on a daily basis now, is to help customers either to design their experiment or completely to just provide them with design of experiments and help them analyze them to help them get the most out of our product. So what is design of experiment for those of you who are wondering? So if we take the National Institute of Standard and Technology definition, design of experiment is a systematic, rigorous approach to engineering problem solving that applies principle and technique at the data collection stage so as to ensure the generation of valid, defensible, and supportable engineering conclusions. In addition, all of this carried out under a constraint of minimal expenditure of engineering runs, times, and money. So by this definition, you can see that design of experiment is a widely used field. It doesn't just apply to our gene therapy industry, but it does answer everything that we want. It's a scientific approach that helps to really optimize our processes, and it's also answering the need to move forward faster and saving money in the long term. So that's everything that we need to apply in our industry to move forward to better results and bring new therapy to the market. So before moving to design of experiments, there are some important considerations to get. So there are three key principles that have to be followed for anyone that wants to do design of experiments. So first of all, everything has to be randomized, meaning that to ensure the validity of your result and avoid any unwanted effects that could muddy your interpretation, we will work with a randomization of every experiment that is done. So that really allows to eliminate all effects that are unknown or uncontrolled in our experiment. The second one is replication. So that's something that is quite standard now in the industry, but still anything that is done with DOE, so it will be an iterative process. So any experiment that gives a prediction will need to be replicated just to ensure that the model that we built out of DOE are correct and just to ensure everything is valid. And the last one is blocking. So it's difficult to get its head around it when we don't know what DOE is, but basically sometimes, and we know that it's the case in our processes, you cannot randomize every factor. It can be simply impossible or just too costly. So you do have a solution with that in DOE, which is blocking, where you basically restrict the randomization by doing some trials with one setting of the factor and the other setting. A good example of that is, let's say you are limited in the number of bioreactors you have. You only have 12 that you can run at once in small scale bioreactor, but you know that your experiment will require to do 24. So then you can introduce blocking, which is basically saying, okay, I will do 12 on one day and 12 on the second day. So there is an actual difference between the two runs, but you can describe that as one block and for the first 12 and the second block on the second 12 runs. That will allow you to still be able to do DOE and do more experiments and have better results in the end without being restricted by the number of bioreactors that you have. So why would you want to move to design experiment? What does it bring compared to a traditional methods that we are used to do? So first of all, DOE will really bring a better understanding of your process. You will test out different factors and you will be able to eliminate the one that doesn't have a significant effect on the response that you study. But also the most powerful aspect of theory is that it will really allow you to understand the interaction and the nonlinear effects of your results. That will really allow you then to predict what result you will get without having to test an infinite amount of conditions. And once you do that, once you understood your process, you will be able to actually optimize it. So you will be able to push the system to the limits, actually explore settings that you may not have thought of, but that the DOE allowed you to show that you should move into the direction and test that kind of thing. This also allows you to look at a multitude of responses and actually pinpoint the sweet spots that answer your question. Most of the time, we are looking at the titer, the VG titer, and that's what we want to maximize. But that's only one part of the whole process. So you will want to look, for example, at the percentage of full capsid or the quantity of impurity at the end of the process, or you want to include some cost of goods decision in that. And using DOE, you will be able to implement that to make a decision based on this criteria that will give you the optimal sets of conditions to reach that goal. And finally, what DOE does as well is, since you modelize your answer, you are able to understand it. You can also start to do robustness testing of your process, which is really the next steps for moving toward a larger scale. So that will allow you to determine your normal operating range on each crucial factor that you will have. And if you don't that, honestly, scale up afterward will be much, much easier once you understand what happens, because scaling up comes with its own constraints, and there are things that you will definitely need to change or to tweak compared to your process development studies. And having done theory at that process development step will really solve your scale up afterward. But how does it work? So a successful theory always follows a design that has been thought to answer one specific question. And this is this design that will determine the number of experiments you run. For example, a really simple design, if we look at just two factors, the quantity of DNA per million of cells, as well as the quantity of vector VAAV per million of cells during your transfection. So you can use a two-factor full factorial design, which will basically be comprised of 4 points, testing the most extreme condition of each factor. So from that are usually determined as -1 to +1. But if we take, for example, DNA per million of cells, that would be for vector VAAV, for example, 0.5 microgram to 2 microgram. And on top of that, you will test maybe a center point that is your current best working condition. And this will bring you to to 5 points in total. And by running this 5 points and feeding that to DOE software, what the software will do is that it will fit a model, a mathematical model that takes the form of an equation as presented there, that will modelize the response within that design space, so within this square. So we'll be able to predict, without having done any experiments, what happens inside that square for conditions that you didn't test. Now, one limitation of this type of design, for example, which is a standard design, but a really simple one, is that the more factors you have to analyze, the more experiments you need to run, because a full factorial design is 2 to the power of x being the number of factors.
So that's only reasonable for 4, 5, maybe 6 factors, but not more than that, depending on your experimental capacity. Luckily, there are obviously approaches that allow you to deal with numerous factors and to still have design that will allow you to answer your responses. So I've been talking a lot about factor and responses, but what does it mean and why is it important? So this is a question that we often get at PolyPlus for people that want to start DOE. What are the factors that we recommend to look at when using DOE in transfection setting? And there are obviously many. So you can look at the quantity of DNA, the medium that you use, the complexation time, the complexation volume, the Victor-Vir to DNA ratio. So that all is linked to really the transfection itself. But you can also look at your plasmid ratio, which can have a drastic impact on your titer and your fluency. But you can also use look at process parameters such as pH, DO, or temperature. So basically any factor you can feel you can test in theory. But what I wanted to show there is that these are the ones that we know impact the titers. And within this factor, depending on what you looked at, but if we think in terms of titer, I think the first one I always recommend to test are the 3 most important, which are cell density at the time of transfection, DNA amount per million of cells, and Victor-VAV to DNA ratio. And after that, we can move maybe to plasmid ratio. And usually, most of the people already have a fixed cell line, a fixed medium. So that's things that have been selected before testing from the transfection, and there are other considerations that play there. And then you run the experiment, and obviously you want to look at your response. And as I was saying a little bit before, so you can do DOE and only look at the VG titer and take your answer and say, okay, I want the maximal titer. But that will be really be looking at just one little amount of what you really want to be looking at. So basically, the more responses you are able to follow and to add to a DOE experiment, the better your decision making will be in the end afterward.
So, and with that, I conclude this part of the talk with just 5 advice I give to customer work with on DOE. The first one is maybe the most difficult one, and as it's not directly linked to DOE per se, is really to master your analytics. And by that, I know it's not always easy, but since we are still working with biologics, we know there is inherent variability already coming into place. And the last thing you want is to not be able to analyze your results, DOE or not, because you already have a 50% variation due to your dPCR protocol, for example. So the first step before going any further will be really to work on validating those analytic process. Then, once you have that, you can really start thinking about DOE, and you will start by determining the goal of your experiment, depending on where you stand within your process development. So in the beginning, you will look at maybe more factors than less, because you would like to screen a little bit, understand better what happened in your process, and the more you move along the way, the more you will be able to move towards optimization design with maybe more complex but less factor into it. This goes right into the third one, choose the right design for your purpose. So there are a lot of variety of design. There can be complete design or partial design. There can be optimal design that are computer assisted. And depending on what you know on your process and what you are confident doing, you can choose the right design for your purpose and minimize the number of experiments you need to run to actually answer your question. And then once the day arrives to actually run the experiment, so for people that never did DOE, my advice really is to take a step back and before going full head into preparing flask and all, really organize your lab and organize the way you are going to do that, because doing maybe 20 to 30 different transaction mix can be a little bit overwhelming, and you don't want that to impact your results in the end. And once you actually perform your DOE and you get your results, it's always a standard practice and a good practice to iterate and confirm. Basically, implementing, expanding your DOE, doing more DOE run, and confirm every result that you get, because it's still just a mathematical model and not the reality. So you want to be sure that what you see with your DOE is actually true. And with that, I switch to presenting us a few case studies.
So the first one is just a successful customer project I had with a customer. So it was not AV, but antiviruses, but basically it's the same. How does it work? And it was really people that wanted a quick yes, no answer. Are we able to reach that titer, and is our process viable for further development? So they came to me, and I provided a first DOE design that was a more screening design. So there was 5 factors. They did 25 experiments. We had broad ranges. The goal was just to see what are the factors that are important and how can we improve. Is it possible to improve the titer? This first experiment gave an interesting results, but we saw that we were actually too broad. So we needed to look at a smaller design space to really be able to see where we can push the response. And all factors were important, so we kept them, but we did more experiments in the second round with smaller range, and we were really able to pinpoint the exact condition where the titer will be maximal. And after that, from the initial process, we actually had a threefold increase, so we were really happy to see that, because it was really difficult to produce vector, and the customer, by using this DOE and having this threefold increase, was able to move forward and say, okay, this process makes sense, it will be viable economically, we can move forward, and they were really happy with it. The second example is something that we did internally at Polyplus. So you may have heard about, we recently launched P-Helper plasmid for customer for AAV. And when we tested this helper plasmid internally, we had great results. We were happy with the first result we were seeing. For most serotype, we did see an improvement compared to the competition, but there was one problem. There was one really important serotype for which we seen really less improvement than with other serotypes. And the thing is that during this process, we mostly use standard plasmid ratio, nothing fancy. We didn't do any optimization of the plasmid ratio itself. And our hypothesis was basically with this new helper, the plasmid ratio that we were seeing was completely suboptimal for that serotype. So we use what we call the mixture design, which is a particular type of design, because the 3 factors that you use, the sum of this factor will always be 1. And this is actually really adapted to a ratio, such as the plasmid ratio. And using the design, we were able to pinpoint that we were far off the best plasmid ratio to get the best titer and best full capsid for that serotype. And I leave mic to Andy to follow with the Endelin case study.
Thanks, Paul. So yeah, so as you know, we applied the principles that that Paul outlined earlier with, you know, identifying factors and nailing down analytics. We look to, you know, apply these things into our platforms and how they impact the performance of those platforms as we develop and optimize them. So, you know, we look at multiple things on the upstream side, like cellular factors, DNA factors, media, transfection reagent, et cetera. And then these things lead directly into and influence the purification strategies that also, you know, have DOE shaped around them with clarification, ultracentrifugation, and diafiltration versus chromatography, et cetera. So, you know, taking these into consideration and applying them, looking at the upstream manufacturing, we look at multiple different factors at multiple different levels and then test the responses. And it's, I can't stress enough how critical it is to not just focus on productivity. It's perhaps the most critical response to look at because the other quality attributes tend to follow suit. But I think I'll demonstrate it a little bit here that it's not always the primary that drives ultimate productivity going forward. So, maximizing viral titer can, have pitfalls in and of itself. So, looking here at productivity and maximizing with the factors that we described before with, cell transfection density, total DNA volume, we come up with response contours that, like Paul was saying, for his P-help, put us in the best space for success in maximizing viral titers going forward. So taking this and the top candidates for productivity moving forward, and also considering percent full capsids because that very directly influences downstream performance. We select and move forward and reiterate and scale up into leader. So that's what we're looking at here. We're looking at the DOE predicted titer versus the actual titers that we've, experimental titers that we derived at a 2-liter scale. So you can see candidate number 2 here didn't have the highest predicted DOE titer and didn't have the highest experimentally derived titer when we actually scaled it up and manufactured it. However, in downstream, it resulted in ultimately in the highest yield in terms of percent recovery and overall yield because of multiple attributes that we were looking at in the upstream with percent full, overall purity. Feed stream is very critical going into downstream. So you may sacrifice some initial titer, but in the end, you end up with more product in general. So here's just a gel that just demonstrates, the level of purity that we're seeing across all 4 candidates. So, we do initial DOE, prove it in scale up into 2-liter, and then once we are into 2-liter, we can show that we have scalability across, or scalability and consistency in that scale from 2-liter to 50-liter. So here you're looking at, consistency in titer, consistency, linear increase in scale, and then through in recoveries through harvest, where you're looking at 2-liter, 50-liter harvest, where we're actually able to increase, in this case, able to increase titer and increase total final yield with a highly pure, high quality product at the end of the day. So then, continuing to carry this out with a different product, that, it demonstrates the point that when you build a robust transfection upstream and downstream purification system that's optimized for a given product, we ultimately see very linear scalability from all the way from 125 mil flask through amber 2-liter and into 2,000 liter. In fact, we're fairly confident. We've showed this a number of times now that once we get into 2-liter scale, we're very confident scaling beyond that into 50, 200, and all the way to 2,000 liters. Expanding this even farther, looking at broader aspects like transfection reagent, we worked with PolyPlus to look at Effectivir versus historical PEI across multiple serotypes and multiple media formulations. And what we found is the Effectivir was a superior product and yielded consistently higher than PEI across multiple serotypes and eventually across multiple scales. So what have we ultimately done here? We've built a platform that is completely modular, that allows for multiple different upstream modalities, whether it be suspension or adherent. And in terms of the adherent, either planar or three-dimensional, and multiple downstream modalities that allow us to fit the process to the product, rather than continuously trying to pound square pegs into round holes. So we have options with all column systems, or empty full separation through ultracentrifugation, multiple steps to get through dialysis and final formulation, clarification on the upside or in the upstream, and also allows us to optimize our downstream recoveries based on those modular designs. We're not stuck with just one pathway. So, what it boils down to is really a simple math exercise. If, we have a campaign where we're trying to target a yield of 2e17 viral genomes, which is, not at all scary in terms of the overall yield requirement. It's very easy to do in a scale out sense, where if we're able to get 2e17 VGs in our upstream with a 20% recovery in our downstream, all we need to do is scale this out to 5 sublots to achieve that final purified bulk yield of 2e17 VGs. And that's a 10-week process, 5 sublots, and all of the cost of goods associated. If we take that 10-week process and we can, through DOE and optimization, have a realization of a 2-fold increase or a 2.5-fold increase, a very modest increase in the upstream yield, we're able to take that 10-week process in 5 sublots down to 2 upstream sublots, which is 5 weeks and a 50% reduction in cost of goods in time. Take that to the downstream, where if we can increase our recoveries by 2-fold, now all of a sudden we're at a 40% recovery. All we need is 1 upstream sublot, 1 downstream purification, and we meet the target yield in 3 weeks, and that translates to a 70% reduction in time and costs of goods. So I think it becomes plainly obvious that a small investment in upstream development can pay large dividends in the end. So having said that, I will turn it back over to Paul to say a few words about leveraging partnerships to expedite process optimization.
Well, thank you, Andy. And it was really interesting, this little math experience, and actually a really good example on how leveraging both design of experiment, but also newer generation of reagent of solution can allow us really to increase the total yield, but also drastically reduce the cost of goods and the time to market. And that's where Polyplus and Anolin partnership makes sense. And that's what we can propose to our customer is our expertise and our DOE knowledge to really improve the efficiency of your process by really being able to predict optimal condition, both for the upstream and the downstream. But also allow you to save time, to invest a little bit more maybe in the process development, but along the way, in the end, you save time on your production and you save money because you need to make less run. And with that, I think let's conclude our talk and we can move to the Q&A session. Thanks you all for your time.
All right. And with that, Paul, Andy, great presentation, guys. Some really, really interesting stuff here so far. And we've got a few questions coming in from our audience already. And audience members, we do have some time now. So if you do have any more questions, please feel free to submit them. We're going to get to as many as we can right now. And any that we don't get to, I'm going to pass along to our presenters today for potential follow-up after the event. Just a quick couple reminders about your interface. That Q&A box is on the right side at the top. Should be highlighting on your screen right now. Below that, there's a couple resources on today's topic you can feel free to download. And on the left side, if you do need to reach out to Andy or Paul with any questions, email addresses should be there, along with my contact information as well, if you do need to get in touch with us for anything else. So with that, guys, let's get into some questions here. The first one came in early from Carlos here, asking, based on the DOE results, which parameters that had a large impact were the most surprising?
So I'm going to start with that one. So on Polyplus side, on my side, and on what I've seen, we're working with customer and helping with DOE. Maybe it's not the most surprising, but I think it's still one that is the most overlooked is really the plasmid ratio. It really can have a drastic impact, and I feel like a lot of people tend to overlook this or just look at a few plasmid ratio that they found in the literature and apply to the system. And really in your system, how the plasmid ratio impacts your titer, but not only your titer, actually also your other parameters is really important. And what you will find in terms of optimal plasmid ratio in the end will most certainly surprise you and be quite far from what you've been using before.
So yeah, actually, I would say the same thing. I concur. It's the plasmid ratio tends to have a large impact on not only overall productivity, but also other quality attributes of the product itself, especially empty-full ratio, and as well as some other quality attributes. And also the surprising thing is how much it changes from vector to vector, depending on serotype and transgene. It can vary quite a bit. How much, what is optimal in terms of the ratio.
Awesome. Yeah, thanks, guys. Let's see. Coming up to our next one. So the case studies that you talked about, they showed that DOE demonstrated some consistent results in terms of scalability. Is that something you'll typically see?
Yeah, it does. I guess I'll take this question. It does lend itself to scalability. Usually with the upfront investment in DOE, we've seen very consistent scale ups all the way, starting as low as 125 mils and all the way up through 500 and 2,000 liters. But yeah, it definitely lends itself to consistency and scale up.
Awesome. Thanks, Andy. Let's see. Next question. Can you estimate empty full ratios at harvest as part of a DOE?
So this is a bit of a challenge trying to analyze empty full particles in a complex matrix right out of harvest, as you can imagine. But there are a few techniques. Capsid ELISA is one technique that's fairly simple and straightforward, but it has it does have some limitations to it in terms of variability. We're looking at other methods to quantify that and characterize that are more robust and have higher specificity. So yeah, more to come on that. And that's something that definitely we're working on to improve.
Awesome. Yeah. Thanks, Andy. A few more coming in here. So let's do this one next. So someone's asking, can you explain a practical application of a modular approach to your platform?
Say that again. I'm sorry.
They're asking, can you explain a practical application of a modular approach to your platform?
Oh, yeah, absolutely. So there's, a lot of different indications, a lot of different transgene, and a lot of new serotypes that are being developed, engineered serotypes. So, for instance, if scalability is an issue, if this is a large indication, then we can work to go through an all-column downstream process. But in some cases, that would be a suspension, large-scale suspension, 500, 2,000 liter in the front end, and downstream all-column purification. But we've also had instances like with ocular indications or ultra-rare indications where things may not go into a commercial setting. They may stay in phase 1-2. Speed is of the essence. At that point, scalability isn't as big of an issue. Speed is key. Adherent systems can move faster. There's less nuance in scaling. It's more of a scale out. So we can move much quicker with that. And also, you may have an engineered serotype that's not well characterized in terms of purification. So downstream applications that are less scalable, like ultracentrifugation techniques, might be appropriate there. So, and there's any combination thereof, and we've seen and done them all.
Awesome. Yeah. Thanks, Andy. Let's see our next one. So it seems like 2,000 liter scale has lower titer compared to other smaller scales. What do you think is the main reason?
That's a really good question. And honestly, it's because when we got to 2,000 liters with it, that we really challenged the system, challenged the process. First of all, it was in a 2,000 liter. It was not 2,000 liters. It was 1,000 liter in 2,000 liters. And bioreactors like to run full. They tend to. It's less efficient to run them at a lower scale. We also introduced a few intermediate steps in the process with some filtrations and things that were suboptimal. So, as a result, you saw a lower titer overall, but our bar was 3 to the 11. So I think, honestly, I'm most proud of that one, because even though we really challenged that system as much as we possibly could, it still met the bar, which really, showed how robust the process truly is.
Okay. Awesome. Yeah. Thanks again, Andy. We've got just a couple minutes here for one or two more. But audience members, if you did have any questions, keep submitting them. I'm going to pass those along if we end up, running out of time here. Let's see. So our next question: have you applied DOE approaches to multiple cell lines?
Yes, absolutely. So we've done multiple DOEs around multiple cell lines. We have our own proprietary cell line that we use. That's probably has the most characterization around it. But yeah, we've done DOEs around other AAV producing cell lines, as well as some other cell lines like Vero and things that are different platforms, even than the triple transfection platform that is the mainstay. So we're looking at multiple upstream modalities in cell lines that DOE applies to almost anything.
Awesome. Thanks, Andy. And I think this is going to be a great place for us to wrap up. So with that, again, Paul, Andy, thanks for joining us today. This was a great presentation with some really interesting stuff. I think we all, learned a lot, and some really fascinating results that you guys are getting. And so with that, audience members, again, thank you all for joining as well. We hope that you have a great rest of your day, and we'll see everyone next time. Bye, everybody.
Bye, thank you.